Well, it's great to be here. Can you hear me? Can we turn it up? Okay, great. So thank you so much for inviting me. This is an unbelievable collection of talks and people, and I'm really looking forward to discussions over the next several days. So this is the cover of a book that the MIT Press will publish later this spring, and it really tries to give a perspective on what's happening right now. What's really going on right now is an explosion. It's a revolution. And of course, during a revolution, there's chaos. That is to say, it's not quite clear where things are going or how things will sort out. And that's what makes it exciting. But also is a cautionary note. We really don't know. And there's a lot of questions here, big questions. So I'm the president of the Neural Information Processing Systems Foundation that runs the annual NIPS meeting. The 30th annual meeting took place in Long Beach last December. 8,000 attendees. Now, when NIPS started, it was in Denver Tech Center. A few hundred people showed up. It was early days of neural networks. And it was exciting because there was a lot of interest in this new architecture that was bio-inspired. And when at the beginning of the meeting, I welcomed the audience. I asked how many people here for the first time. Half the audience puts up their hand. A lot of young people out there. It's been increasing by 50% over the last 3 years, and that's exponential. It's not quite Moore's law, but we'll see how longer that lasts. But here's how I introduced the meeting to the audience. I said, 30 years ago at the very first NIPS meeting, little could I have imagined standing here in front of you and addressing 8,000 people. I thought it would take 10 years. And that's because, of course, we were optimistic, and it wasn't the first time that academics were optimistic. If you go back to 1956, which was the seminal meeting at Dartmouth that kicked off artificial intelligence, it was also a very exciting time. Computer programs were being written that could solve theorems in mathematics, that could play games. And it was felt that intelligence was just around the corner. We just have to write a program that could replicate the behavior, the intelligent behavior of humans. Now, again, little did they know how complex that problem would be. But nonetheless, off they charged. And Marvin Minsky, who was one of the founders of AI, when he started the AI lab at MIT, the first DARPA grant he got the project was to build a robot that could play ping pong. Now, little did he appreciate how difficult that was. And we still don't have robots that can play ping pong as well as humans, right? But here's the story. The story was that when they got the grant, they realized they had forgotten to ask for any money to write a program for vision. And so they assigned it to a graduate student as a summer project. Now, 50 years later, there was another meeting at Dartmouth, 2005, that I was invited to, and the idea was to look and see where AI is and where it's going. That was a very interesting meeting. And I had a chance to ask Marvin, is this true? And he said, you got your facts right. We did not give it as a summer project to a graduate student. We gave it to undergraduates. And there's actually proof of this. So this is a AI memo that was, as you can see, 1966. This was a summer vision project, and the idea was to get a bunch of undergraduates, give them all a project like you do segmentation, and then at the end of the summer we'll put them all together, and that'll be the vision program. I mean, it was really very naive, and here's the reason why. The reason is that nature took hundreds of millions of years to evolve efficient vision systems. Not an easy problem. Half of our brain, humans, is devoted to vision. And it's so easy for us. We open our eyes, we see objects, right? It seems like it's going to be a trivial problem. And that's because there's so much hidden under the hood that nobody could have expected, or at least anticipated back then. And so here's why vision is a hard problem. So these are zebra finches, and it's obvious to you that they're the same species. the traditional approach in computer science back then was to do template matching on the pixels. And you can see that's going to fail here. Because if you have two different poses, all the pixels are going to be different. Well, of course, the solution that computer vision hit on was to look for features. And now became a very long-term, decades-long project to try to characterize the characteristic features that it can discriminate between different objects. And that turns out to be very difficult because there is no unique feature that distinguishes each object. There's combinations of features, and features are shared by different species, by different objects. And so therefore, the way you make progress is by spending a year or two trying to analyze a particular category of objects, like cups, and figure out how to discriminate them from everything else. And with that approach, which is very labor intensive, you can make progress, and it was in computer vision about a 0.5% reduction in the error rate per year. And you know, that's progress, and it was steady progress, and it was clear that that was, eventually would pay off at great expense of graduate students. Okay, so there's another approach that was around the same time. Frank Rosenblatt realized that, one of the important features of the human brain is that it's massively parallel, and there are these relatively simple processing units. We now know they're not simple, but you can simplify them as a summing device of a lot of inputs. You can think of them as pixels weighted and then summed and then thresholded, right? Now this is actually not, this is the simplest possible neural network model. In fact, this is a single neuron model. It's a single neuron, and the question is how much computing can you do? Well, what made this a really attractive approach was that he came up with a learning algorithm which automatically changed the weights every time you gave it an input and then categorized it. If you made a mistake, you changed the weights to improve it. And he showed that if there's a set of weights that could solve the problem, the learning algorithm was guaranteed to find it. Now the problem, the fly in the ointment here is that as Minsky and Papert showed in their really excellent geometrical analysis book published in 1969, that perceptron is only capable of solving linearly separable functions, first-order predicates. And that's a real limitation because most real-world problems are more complicated. And then they showed, and I don't know why they put this on the cover, it turns out that one of these two is a completely connected curve, and the other one is two separate curves. How many of you know which is which? Right? This is a problem that the perceptron fails on. It turns out most humans do too. It's really like, it's almost a joke. But no, actually, the problem was that after this fantastic analysis, and it's really worth going back and looking at it, it's really a beautifully written book. At the very end, they say that in their view, in order to be able to make progress, we need to have a learning algorithm for multi-layer perceptrons. And in their view, there would not be ever discovered such an algorithm, and this was a sterile direction to go. Well, okay, it turns out that this stopped research funding jobs for the next several decades. It was, it ended the whole process. And by the way, it was really interesting. Frank Rosenblatt, because computers were so slow, he actually had to build an analog machine that had motors that turned potentiometers, right? I mean, this was really a very a pioneering time when he just was doing basic concepts. It was clear that the hardware was way far behind, and it was difficult to make progress. But I have to thank Minsky and Papert because they made my career possible. And that was when I met Jeff Hinton. So Jeff and I basically come from very different backgrounds. He comes from psychology and AI at the time, and I was coming from physics, theoretical physics, and neuroscience. But together, we mind melded, because we basically believed that the solution was to look at the way the brain is put together. And our reasoning was very simple. It's the only existence proof you can solve these problems. So why not look inside and see how it's put together? And within a couple of years, we created a network model called the Boltzmann Machine. And I'm not going to go into any details. This is back in the 80s, except to say that there was a learning algorithm that we discovered which solved the problem of learning in multi-layered networks. We had hidden units. In fact, we named them. This is where the term hidden unit comes from, this paper. And the dam broke. And it turns out there were dozens of learning algorithms that could solve the multi-layered. It wasn't just, was unique or anything. And in fact, Jeff, together with Dave Rummelhart, came up with another more efficient one called backpropagation that really has proven the test of time. But it took a long time to go from proof of principle, simple networks at the time, because computers, if you just think about them, the VAX class of computer was megahertz, right? And now we've increased in computer power by a factor of 1,000,000. Data has improved with the internet by a factor of 1,000,000. And at a momentous meeting that was held, NIPS meeting in Lake Tahoe in 2013, Jeff and his students revealed that they had a deep learning net that could decrease the error rate of ImageNet by 20%. This sent shockwaves through the computer vision community, because if you're going at a 0.5% per year, that's 40 years of research. Suddenly, and more importantly than performance, it didn't require much human labor. It required a huge amount of computation. The trade-off here is between whether you're going to use human labor of an engineer or you're going to use computer power. And at this point, it's crossed over. Computer power has gotten so cheap that you're much better off with the learning algorithm. Not only that, but the very same learning algorithm could be used to solve many problems. Whereas if you're writing programs, each problem requires a different program. And if they're very complex problems, they require huge programs. And of course, that doesn't scale. Okay, well, how do they go from it's 30 years, 1983 to 2013? It's 30 years. What the hell was happening over those 30 years? And here's what happened. They looked into the brain. We looked into the architecture of the brain. And it came from work that was done earlier in the 1960s by Hubel and Wiesel. Two neurophysiologists who put an electrode into the cat brain, recorded from single neurons, and for the first time reported that unlike the retina, where the receptive field is the region where you get a response is circular, in the visual cortex, and here's a cortical neuron pyramidal cell, here's a microelectrode, tungsten microelectrode, they reported that the cells there were oriented, and all orientations are represented everywhere. This is a simple cell that had a region that was excitatory and adjacent to a region that was inhibitory. And this is repeated over the entire visual field, dedicated hardware in primary visual cortex. That's a convolutional solution. So the principle in V1 is convolution. Many, many features are represented. But simple cells weren't the only ones they reported. There was another one called the complex cell, and it was distinguished because it didn't have subfields. And in fact, responded to an oriented bar, as you can see here, regardless of where it was located within this patch, the receptive field. And as you can see, it's oriented. They called it a complex cell. And it's clear now what it's doing. It's computing an element of invariance. It's able to generalize across of a particular feature across a small patch. And then, of course, it took a long time for neuroscientists to go one neuron at a time and climb the entire ladder of the visual cortex, 24 areas containing neurons that respond to visual stimulus, and realize that as you go higher, the response properties become more and more specific over a larger and larger part of the visual field. This is a hierarchy. And we didn't know it at the time, but we know now that if you have a given fixed number of hidden units, you're much better off putting them in a hierarchy than having a flat representation. And that's what happened. Jan LeCun, who is really the powerhouse behind this particular architecture called convolutional neural networks, which is the most popular and successful of the deep learning algorithms, realized that together with Jeff, this was taken at a meeting of the Canadian Institute for Advanced Research, where I was on the advisory board, that you could have many layers of these convolutions, and they're very cheap because of the fact that the weights are the same across all the filters, of course. And then at the very top, you could do an all-to-all. And basically, it was incremental advances. Year after year after year, they improved it, more and more data, and so forth. And what they ended up doing, and this is the critical point, was that they realized that there was a common theme for each of these layers that produced the best results, that you first start with a linear filter, you threshold it, nonlinearity, and there's various types. The one that's used now is rectified linear hinge function, and then you pool. Well, this is like a simple cell. This is like a complex cell. And then you normalize, and it turns out the visual cortex has inhibitory cells, feedback, which helps you to normalize so you're within the operating range. So all of these things actually are represented in the brain, and Jim DiCarlo, who actually has recorded from each of these locations, has shown that the properties of cells all the way up have statistical properties that are identical to those that are found in the visual cortex. The difference now is before, we didn't know what the properties, why do they have the properties they have? Why do you have a hierarchy? We didn't know that. Well, we have a computational solution here that explains why. And that really means that we have a much deeper understanding in neuroscience now than we had before. Now, going back to the 80s, you have to understand how naive we were. So, statistical models at the time had, fewer than 100 parameters. And statisticians basically spent their whole time, proving theorems about convergence and things. And here we were, with networks that had thousands of parameters. And NetTalk, which is very famous for being able to convert letters into sounds, pronunciation, had 10,000 units in it. 10,000 dimensional space. They thought we were loony, that we were going to overfit. And did that stop us? No, we just barreled ahead, and it seemed to converge, right? Now, the other thing, of course, we were told was that, this is by the optimization crowd. Stochastic gradient descent will never work because there are these local minima, and you're going to get killed. You're going to never reach the optimal solution. Well, we just turned it on. Every time we ran it, we got a different solution, and they were equally good. And fast forward, we now understand the math behind why we were successful. And the reason, at least for the learning part, is that if you're in a very high dimensional space, and now the spaces have billions of dimensions in terms of the number of connections, you're always dealing with saddle points. You never have a local minimum. It's very, very rare. And it's because our intuition is based on 1- and 2-dimensional spaces. And that's the problem. Our intuitions are really limited by our experience. And it's not until you go off into the frontier and see what's there, you'll never be able to make any progress. In any case, I don't want to talk about old glories, I want to talk about new ones. And we all know that last year, DeepMind developed a program that beat the world's champion Go master, Qi Ji of China. By the way, this was a tectonic event in Asia, in China. This little kid, 19-year-old kid, is a superstar. He's like, LeBron James. Everybody knows who he is. He represents a very important part of their culture in Japan, in Korea. And that's, for them, it was a much bigger event for them in terms of their emotional response than it was here. Okay, it's another game, right? But how did they do it? Okay, it turns out that deep learning is not enough. Deep learning is basically a very simplified model of the cortex. But under the cortex is this incredibly complex brain that's running the whole thing, running the show. And one of the most important parts is called the basal ganglia, where we now know that there's a reinforcement learning algorithm that learns not by getting detailed feedback, but simply by whether you got it right or wrong. And coupling a reinforcement learning called temporal difference learning with deep learning was the magic sauce that solved the problem. Now, the problem, is in some ways very artificial, complete knowledge. There's no uncertainty. The real world is filled with uncertainties. The real world is very much more complex, despite the fact that humans, really had to put a lot of effort into winning the game. The fact is that any child can be able to navigate the complexities of the world, right? So let's take an example of the real world. And I want to show you what might be possible in the near future. And this is an example from a colleague of mine who at the time was at UCSD in a Science of Learning Center funded by NSF. And our goal was to try to improve education. So Javier put together a social robot called RUBI. And this is one of the first generations, and this is the most recent generation. And he took it into a preschool. These are 18-month-old toddlers. Now, if you talk about uncertainty, this is a chaotic situation because these little kids are running around. They pick up a toy, they have a very short attention span, they throw it down, they run off, and they do something else, right? And it's just, this is really the ultimate. You know, if you're going to want to attract their attention, what do you have to do? Now, when they first put this robot in, and you know, it has arms that can grasp things, it has eyes, expressive eyes and face, and it has cameras so it can look around and it can recognize faces. The first thing that happened is the boys ran over, yanked the arm off. Well, of course, what do you expect boys to do, right? And so went back to the shop, and you know, it's putting in an industrial strength arm is not a solution because you can't have that in a classroom, right? So here's what they did. They had a pressure sensor, you know, cost them $2. And when it went above a certain threshold, RUBI would cry. The boys would back off. The girls would go and hug him. It's called social engineering. And really, the real issue is how do you interface with a human? What counts as an interesting experience for an 18-month-old? And it turned out that the secret, and this is something we'd never anticipated, and you only do it by going and trying it out, is that if a child comes up to you and hands you something or points to something, if you don't respond within a second, you're toast. They'll lose interest in you. And so the key is this little window from about a half a second to a second and a half. But if you hit it, magic happens. The kids love it. They'll just go on for hours pointing at things. And you can really engage them in a way that really went way beyond our expectations. So there's so much out there in the real world that we can really learn from by putting experiments out into the real world. Okay, well, that's another completely different story that I'm going to stop here, except to say that I think this is already beginning to happen with these smart speakers like Alexa, where now the voice is being used to interact with humans. And this is really a new era. I'm going to go out on a limb here, make a prediction. You know that keyboard you have that's on your computer that's 100 years old? Okay, it's going to be in the museum within the next 10 years with the typewriter. Okay, don't hold me to it, but because a natural interface for humans is precisely speech, vision, and now we have the interfaces for actually being able to do that. Okay, well, this is a neuromorphic engineering crowd. I want to say a few things about that. And again, I want to go back to this theme, is that you want to look at nature, you want to extract principles, not details, and then use that to inspire hardware. Really, the future is hardware. You know, these learning algorithms and everything have been around for 30 years. What's new is going to be the algorithms that are going to be implemented, and then the exploration really begins because now you can really scale things up. To something that is closer to the size of the brain. Let me give you one example, a little data point. So the biggest deep learning networks today, they have about 1 million units, like neurons, and about 1 billion connections. Well, that's roughly what you find in a cubic millimeter of the cortex. And the cortex has about 20,000 cubic millimeters, 10 to 20,000. So it just shows you how far away we are. Now, the problem is that if you have the specialty, and each part of the cortex is specialized for a different task, right? But if you have all these specialists, how do you organize them? That's an unsolved problem. Global organization. Maybe that should be one of the things on our agenda. Okay, well, we're interested in vision, right? How do you really crack that problem? I don't think we really are quite there yet. I think that deep learning has done something impressive, but there's so much more that needs to be done. And the way that you do it, in finances, you follow the money. Well, here you follow the photons. So this is taken at Harvard Neurobiology back in the 60s. You've already met these two researchers. This is David Hubel and Torsten Wiesel. This is Steve Hoefler, who founded the department. And it was my privilege to actually do a postdoc with him. So he taught me what I know about neuroscience. Now, at the time that, that they were doing these experiments, this was back when Steve and his lab were at Johns Hopkins University in the Department of Ophthalmology. And Steve was really a synaptic physiologist, knew nothing about vision, felt guilty. So we talked about this during experiments. And so he told me that he felt that he had to do at least one experiment. And so he put an electrode into a cat eye. This is in vivo. For the first time reported that there were cells that had these properties, on-center, off-surround, and the inverse, the off-center, on-surround. And this turned out to be universal in all mammals. And the key to understanding how vision is built up from the outside in. Okay. So what did he do? He said, okay, I've done my duty. This is a classic paper. Everybody knows it. He handed the project off to his 2 postdocs and said, why don't you put your electrode a little bit further up and see how it changes? And that was Hubel and Wiesel. In 1981, they won the Nobel Prize for their path-breaking work. Okay, now fast forward to 2008. Toby Delbruck, who was a student of Carver-Mead, developed this camera, this DVS camera, which was really very simple, but it mimicked the principle of the retina. That is to say, every pixel had 2 different outputs, and if the intensity went above a certain threshold on the on pixel, it would produce a spike, and if it goes down, the off pixel would produce a spike. So the only thing that you get out of the camera are a bunch of spikes, just like the retina. The only thing that comes out are a bunch of spikes. And the beauty is you can actually watch this, and this is now Toby being observed by his own camera. So what you notice, first of all, that most of the pixels are gray because nothing is changing. It's very efficient. There's only a relatively few spikes, but you can see the black and the white, the on and the off, are represented, have a lot of information about motion. So in a sense, you're already encoding things that are important. And also, as you can see, it's insensitive to filters with sunglasses. And it really is a very interesting representation. And just one last example. This is going around roughly every 10 milliseconds. And you can see that in a single frame, for example, a 30 millisecond frame in the camera, it would be just an annulus. But you can see here, you can go down to microseconds. So the timing capability of this camera is really at a very, very high level. That is to say, there's a lot of information that you can get out of it. Now, here's a little anecdote, which is very amusing. This is actually going now down the street in Zurich, where Toby's Institute for Neuroinformatics is located. Toby said he took this to a computer vision meeting. And the beauty is, you can take your whole thing there. It's just a little camera. It only weighs, a few ounces. And he set it up, waved his hand. And people would come there and they'd look at it. And they'd see all these spikes. And then they would say, well, where's the image? And Toby says, no, there's no image. We're just extracting information. And they would say, what does that have to do with vision? Now, interestingly, the people who did pay attention to him are people who work on robots, because with robots, you really have to have low power, and you have to have something that's light. You can't afford a supercomputer on your back, right? So that's really the growing market, and it's really exciting to see how this happens, is that you go from some simple principle, and you can build on that. And this is really what we're trying to do. This is showing how you can very easily segment things in a cluttered scene. Okay, so where are we? 2018. I already told you we're in the middle of an explosion. Well, it turns out there's another explosion going on in neuroscience. Because in 2013, President Obama announced to the world, and by the way, everyone was listening, not just Americans, right? That for his administration, he had picked the brain as his grand challenge in science and technology. And as you can see, for the acronym for BRAIN, the key letters are I for innovative and N for neurotechnologies, because the goal here was to be able to go from 1 cell at a time to 1 million cells at a time by getting engineers to work together with neuroscientists and develop new tools that will accelerate research, regardless of what part of the brain you're studying, regardless of what problem you're studying. If you can collect the data faster, better, cheaper, you're going to make progress faster. Okay? Now, I was involved in this process. I helped bring it to the White House. There were competing projects. For example, NASA wanted to lasso an asteroid and put it around the moon. Space cowboys. Department of Energy wanted to build a better battery, and of course, very practical. But Obama picked the brain, which was really, in a sense, the right choice. Because this is going to yield new insights into how the global brain works. I was telling you before, we don't know how it's globally organized. But unless you can record from a significant fraction of the brain, you'll never find out. And so I'm going to give you one example. And it's, by the way, over the last 3 or 4 years, it has been spectacular in terms of the progress that's been made. Now it's possible to record routinely from 1,000 neurons at the same time. In fact, I'm going to show you an example from my own lab. This is work done by Sophie Ayman, a postdoc, working with Ralph Greenspan, who's an insect neurophysiologist who, as you can see here, this is a fruit fly that's sitting on top of a ball that can rotate. And there's a hole in the head of the fly. And there's a camera on top, it's called light field microscope, that actually can see in 3D. And from that, you can extract information coming out of these different structures. And I'll just point out a few of them. This is called the central complex here. And it has things in it like the ellipsoid body, which is circular. You're going to see this in the movie coming up. There's a fan-shaped body, a protocerebral bridge. And here, by the way, are actually snapshots from the actual recording. These are neurons that have been labeled with a calcium-sensitive dye, so they light up when they fire. Now, what's amazing here is that these different colors actually are false colors, but they've been selected out by independent component analysis. This is an algorithm that I developed for blind source separation back in the 90s. And it's just wonderful to see how that algorithm now is used in thousands of applications, but I'm particularly pleased that it's being used here to dissect out the different parts of the fly brain. And it's been used in many, many other places. Okay, so this is now what you're going to be looking at, and what you're going to see is as the fly is behaving, and this is really behavior in real time, you're going to see different parts of the fly brain as they become active. This is truly magic. So here are the mushroom bodies up here. You see that when the animal's grooming, not much activity, but as soon as it moves, you begin to see some of these structures, like the ellipsoid body here. And it's really clear that when you move, much more of the fly brain gets engaged. So just doing this only takes a few neurons. By the way, there are on the order of 100,000 neurons in the brain. And the complexity is far greater than you would think because of the fact that a lot of the activity occurs inside of the neuropil, where dendrites and axons come together, and not in the cell bodies. So but ICA separates out the hotspots in the neuropil. So we are seeing something here for the first time in history that we could have only dreamed about, even just like 5 years ago. Being able to access the activity patterns in the entire brain. It's been done in a zebrafish, which is a vertebrate. Again, about 80,000 neurons. And it's being done in mice. I'm part of a DARPA group called NESD, which is going to implant little chips that's going to allow us to record from 1 million neurons at the same time throughout the entire brain with very high spatial and temporal resolution. Now, just before the announcement was made in the White House, this picture was taken. This is me, in case you can't recognize me. But these are the people who actually were the prime movers and the agencies that are going to be responsible for moving forward with the brain initiative. And really, the spark plug is Myung Chun here, who is diminutive compared to all these tall, towering males. She's the one that actually got it into the White House, into the president's attention. She is the scientific officer of the Kavli Foundation. Francis Collins is the lead. The NIH is the lead organization, but NSF, Cora Merritt. And this is something that was very deeply embarrassing to me because we're standing here waiting. Before this picture was taken, Obama wanted to thank us personally for helping out with the initiative. And so I knew a lot of these people. I mean, I recognized Francis Collins just by his picture. But, I had no idea who this woman was, so I was talking with her. And I said, well, by the way, could you tell me what you do? And she said, I'm the director of DARPA. And then I realized this is a great place to network. And it was exactly at that moment that suddenly somebody tapped me on the back. And I turned around, and Obama is towering over me, much taller than in photographs. It was really, either that he's tall or I'm really short, you know. And I was so flustered, I wasn't prepared. I didn't even introduce myself. All I did was to say, "Thank you, Mr. President," and "Thank you." Time for questions?
So I have a question. So Terry, I was in your reading class when Jeff Hinton's 2006 paper came out, and I remember we knew there was a cusp of some excitement about training deep networks, but I mean, this obviously is more than I think we anticipated then. So you talked a lot about vision, and obviously that's the driving application for neural nets in general, especially what we see today. The brain, of course, you said human brain's half vision, but it's half not vision. So aside from reinforcement learning, basal ganglia, and so forth, where do you see the sort of next wave of brain inspiration outside of vision? What other cognitive functions should we look forward to?
Great question. So I think we're just scratching the surface. Like I said, we have some algorithms we extracted from the brain for the cortex and for the basal ganglia, but there are hundreds of other structures that are doing important things that are probably needed for autonomy, which is one of the goals, right, is to produce something that is able to survive on its own. By the way, I mean, it's really you heard it described, a deep learning network basically has to be coddled. You know, you have to prepare the data for it. You have to basically watch over it. And then, you know, there's all these hyperparameters, you know, which is still a black art. The reality is that, you know, if this is how nature created, you know, it would never, you would be eaten immediately. Deep learning wouldn't survive a day in the real world. By the way, I have to tell this a very short story. This was back in 1989, when the learning algorithms were just really coming to the fore. I was invited by Michael Dertouzis to give a talk, a distinguished lecture, at the AI lab. And it was the Laboratory for Computer Science, it was called. And, you know, I was really pleased. I arrived, and we had a great discussion in the morning. And he said, well, we have a tradition here. We have a lunch for the speaker, and you'll have a chance to have a discussion with the faculty. So as we're going up the elevator and about to arrive, he said, and oh, by the way, they hate what you do. And we'll give you 5 minutes to start the discussion. And, you know, I said, my Lord, you know, what can I possibly say in 5 minutes? So as I walked over to the food stand, right, there was a luncheon buffet. So I noticed there was a fly. And I said, do you see that fly? That fly can see. It can find food on its own. Not only that, it can fly. And it can reproduce with 100,000 neurons. You have a supercomputer downstairs, this crazy supercomputer. You spend $100 million on it. It can't see, it can't fly, and it's never going to reproduce. What's wrong with this picture? Dead silence. By the way, it was like Daniel in the lion's den. There were rows. The first row or circle around me was senior faculty, and then around them were junior faculty, and then even in the back, you know, the rafters, there were these students, right? And so finally, one of the senior faculty said, well, we haven't written the vision program yet. And, you know, it was a time, strategic computing was a $350 million program at DARPA to build an autonomous tank. And so they had a supercomputer in a tank. It was a disaster. You know, when it came time to actually parade it down in front of the generals, the sun went behind a cloud and it actually charged toward the generals, and they had to run for it. Okay, so good luck with that one. Jerry Sussman said, well, we have a proof. Turing has shown that any computable function can be computed by a Turing machine. And I said, well, the fly in the ointment there is that you have this serial processor, and you don't know how long it's going to take. If you are in a jungle and there's a tiger, you cannot wait for the program to run itself out because you're going to be eaten, right? So time is something that's not an essential part of the whole computing process, right? It's really essential that you actually solve not the perfect solution, but a good solution quickly. And finally, there was a undergraduate student just waiting, bubbling away. I can tell this is a really smart kid. Puts up his hand and says, I know the answer to the question. In fact, everybody in my lab knows the answer to this question. The supercomputer is a general purpose computer that can solve any problem that you program it to do. It can, thousands of programs. But the fly is a special purpose computer. And it can only solve the problems that nature solved for it. And he was absolutely right. And in fact, this is the reason why studying the fly brain is actually the right thing to do. And the reason is that in order to get the most efficient algorithm, you have to actually wire it in, hardwire it. And so the anatomy is actually the algorithm. And by knowing the anatomy, you can reverse engineer it. And so that's what's really unfolded over the last 30, 40 years. We finally, realize that there's a lot to be learned, and much, much, much more because of the fact that there are the superior colliculus that guides the eyes, there's the hypothalamus. I mean, these are all structures that are involved in emotional decisions, evolved in being able to actively seek information in the world. And all we need is to really look at the data, and that will be coming out in reams from the Brain Initiative to see how that's all done. But Hava, you were waiting here very patiently.
Yeah, it's a great talk. Thank you very much. So in the last 3 years, there is a lot of work that is busy with demonstrating different type of deception that happen particularly with the deep and convolutional neural network, to the point that I think 2 weeks ago, Benjo put a paper in the archive where he's talking about the different deception methods and trying to get a conclusion that from his point of view, perhaps these neural networks are not doing what is expected, they are not abstracting, that perhaps are not enough biomotivated to do what we want. So I think they're kind of different camps, the more optimistic, the more pessimistic. And do you think it's a big problem that there are this deception, or do you think it's just a side effect of some lack of understanding?
Okay, so that's I don't have the answer, and if I did, I'd have a grant from you. But what we see in nature is that you can actually do a lot with a little. In other words, that fly only has 100,000 neurons, so I mean, it should be possible, without going to deep learning, to solve the autonomy problem. I think. I think that it's just it's a different class of algorithms. It's really control theory is what it is, but on steroids. But the issue, I think that you're absolutely dead on with regard to the issue of stability and adaptability, is that somehow nature has solved it. But what we don't know is we shouldn't be looking for one solution, the magic bullet. And in fact, I didn't put the slide in, but I usually have a slide that shows the different levels on investigation in the brain. Down here, you have atoms, and up there, you have the whole central nervous system. And there's 10 orders of spatial scale in between, and there's interesting things happening everywhere. There's synapses, there's neurons, there's circuits and networks, and then there's maps. And there are mechanisms at each one of these levels that we can exploit. And someone mentioned dendrites in the earlier talk. I'm particularly high on dendrites. I think that it's doing much, much more than summing. There are nonlinear mechanisms in the dendrite, and the learning algorithms are also very interesting. But there are things going on at the global level. For example, memory consolidation during sleep. Why is it that you sleep for 8 hours? Well, you sleep in part to be able to get the experiences you've had during the day and integrate them into the rest of your knowledge base without disturbing it. Figuring out where the best place is that it belongs, and strengthening it, adding it. And that's a process we don't really understand. We've made progress recently, which came from a study that was done on patients that have epilepsy. And they put in grids of electrodes looking for the focus of the seizure, and they're awake and sleeping, and they're around for a couple of weeks. Well, we studied sleep spindles, which occur during intermediate sleep, and these are 10 to 14 hertz. They last for 2 seconds, thousands of times during the night. And it turns out that they're not synchronous across the whole cortex. There's a traveling wave that goes circularly around from the temporal cortex to the parietal cortex to the prefrontal cortex. We call them Princess Leia waves. And I'll be willing to bet you that this has something to do with the integration of information coming from the hippocampus, which is the sort of the specific store of links that link specific episodes and objects, unique objects in the world, to the general purpose knowledge store. And that's what we need. We need to have a hippocampus. We need to have sleep processing. And these are all things we can reverse engineer.
Thank you.
So let's thank Terry again.